Yeah, we're moving up a concentration gradient there, an electrochemical gradient. So what do I know is happening if we're concentrating acetylcholine molecules in the vesicle? How's it happening? Active transport. And I know some forms of active transport or primary active transport involve the hydrolysis of ATP directly. So if I start using ATP, one thing that may happen locally is I start to generate adenosine as well. If we look inside the vesicle, we find not only is acetylcholine there, but there's also adenosine molecules there. And so when an action potential comes along, say an alpha motor neuron to release acetylcholine at the neuromuscular junction, and we exocytose the acetylcholine, and it's going to diffuse across the cleft and bind to nicotinic cholinergic receptors in the motor end plate, things we'll be talking about next year. At the same time we release acetylcholine, that alpha motor neuron is releasing adenosine, which binds to receptors for adenosine on the presynaptic terminal. And you know what it does? It inhibits the presynaptic terminal. Why would we want to do that? Why would a cell, when it starts to exocytose neurotransmitter, simultaneously start to inhibit itself from exocytosing more?
Just wants to stop it. It's a negative feedback loop to make sure.
It's kind of a negative feedback loop. It doesn't prevent further exocytosis, but it makes it a little less likely. So what it's doing is kind of conserving on our vesicles. We're already releasing acetylcholine. If another action potential comes by soon, release a little less. We're going to conserve on it, so we make sure that if this is the beginning of a whole chain of action potentials, if this is a tetanic stimulus, that we won't run out of vesicles to release with the 100th action potential. It kind of moderates our response. Okay? And that's what I think of when I think of adenosine as an autocrine. Okay, next question then. How do those signal types alter cell behavior? Okay? We're altering cell behavior. We want the cell to behave differently than it did before. And what do we mean by cell behavior then? We need proteins to be acting differently than they did before. So we need to alter protein function. Need to alter either specific activity or total amount. So these signal types in general can be broken into two groups. Whether we're talking about neurotransmitters, paracrines, autocrines, or hormones, these chemical signals are either going to behave as though they're are either going to be hydrophilic or they're going to be lipophilic. Notice what lipophilic I can spell. Hydrophilic, I'm not sure, but that's the way I spell it. Okay, so either they like water or they like lipids. Yeah.
Just for clarification, isn't lipophilic always hydrophobic?
Yeah,
Okay.
so I could say hydrophilic or hydrophobic.
Okay.
I want to show off my vocabulary there. Lipophilic. Okay? So, what was it? Albert Einstein has a quote about the single most important decision you have to make in your life is something on the order, does the universe dislike you, or something like that. And I bought into that. Is the universe kind of mean to you or a happy place? So if we're talking about hydrophilic signals, this determines so much about the signal. Because a hydrophilic signal can't readily pass through a cell membrane. a lipophilic signal or hydrophobic signal, that can pass through a membrane. Now, the vast number of things that we talk about are hydrophilic signals. Okay? Things like the polypeptide hormones, neurotransmitters, most of the neurotransmitters, most of the paracrines and autocrines are hydrophilic. And then lipophilic would be the steroid hormones.
On that, is it including facilitated or active transport?
Not talking about anything other than on its own, can it pass through a membrane or not?
Okay.
Okay?
Okay. I get it.
So when I start learning about chemical signals, like when we start learning about hormones, the first thing I want to know, if we're talking about a signal, is it hydrophilic or hydrophobic? Because I've got this little mental note card for signaling systems. I'm going to tell you that little mental note card right now. I'm going to do it quickly. Have a little mental note card. The moment I know a signal is hydrophilic, I know that it likes water and can't pass through membranes. What does that tell me about a signal such as a polypeptide hormone?
It works on the membrane receptors.
Okay, we're getting things, but I go in order. So the moment I hear something on the order of growth hormone is a polypeptide hormone, here's what flashes through my mind. Synthesized ahead of time, stored in vesicles, exocytosed on demand, freely circulate in the blood, bind to a cell surface receptor, generates a second messenger, which alters specific activity, fast onset, short acting. Bang, 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 bang. You tell me some signal I've never heard of before, and I ask you, is it hydrophilic or hydrophobic? You say it's hydrophilic. I go synthesized ahead of time, stored in vesicles, exocytosed on demand, circulate freely in the blood if it circulates, binds to cell surface receptors, generates second messengers, alters specific activity, fast onset, short acting. Every single time. Okay, steroid hormones or hydrophobic compounds, they can pass through membranes. They don't like water, which is kind of an issue because they're living in an aqueous environment. But they can pass right through membranes. So if you said testosterone and it's a steroid hormone, the other mental note card I go through is synthesized on demand, diffuses out of the cell, needs to bind to something while in the blood, diffuses through the target cell membrane, binds to intracellular receptors, either cytosolic or nuclear, alters gene expression, increases total amount of a protein, slow onset, long-lasting results. And if I really emphasize the slow onset part in my mind, I start to determine or figure out that when we go through and we learn about all the different hormones in our body, the hydrophobic ones, the ones like the steroid hormones, those are regulating things that take place over the time course of days to months to years. Differentiation and development, maintenance of tissue. Okay, when I go to memorize, and so it was important for me to memorize the steroid hormones, and I did it in an organized way. Did you know that if you ever need to go shopping for steroid hormones, there are 7 of them, and you only have to stop and shop at 3 places. First stop, go to the gonads. Estrogen, progesterone, and testosterone. I got 3 out of the 7. I picked them up right there in the gonads. Next stop, I go up to the adrenal cortex. I pick up aldosterone, cortisol, and androstenedione. There's 6 out of the 7. I've only shopped 2 places. Last place, it's a quick trip to go from the adrenal glands to the kidney, where I can pick up calcitriol. The way I memorize those, the first 6 are all made from cholesterol, all have the same T regulatory enzyme, pregnenolone. It's only that 7th one, calcitriol, which is made from vitamin D. If I immediately think, you look at those, the first 3 I had on the list, those are for reproduction and tissue maintenance, tissue development and maintenance. The next 3, one of them is involved in reproduction, and the other 2 are kind of longer term regulation of sodium and osmotic balance in your body and dealing with stress, your body's ability to deal with stress.
What were those second 3?
Aldosterone, cortisol, and androstenedione, which is like a weak sibling of testosterone. Don't have to worry about that for this class. Just want you to know, and I get to teach you about this, but if I'm not here next year for one reason or another, I want you to know that there's ways to remember this stuff to make it easier and have it make sense. Okay? Thyroid hormone. Jingu, you were thyroid hormone, right? Thyroid hormone. Anybody know what's thyroid hormone? Huh? It affects our metabolism seasonally.
It's iodophores.
So what would you kind of guess, huh?
It's an iodophores. It's I3 and I4.
There's T3, T4, number of iodines, yeah. Was it steroid or a polypeptide?
It's neither.
It's neither.
It's an amino acid.
It's neither a polypeptide nor a steroid. But it behaves like a steroid because it produces seasonal changes. Okay? It's made from, thyroid hormone's made from what? It's made from an amino acid. Which amino acid? Tyrosine. Good. See, if you yell at me, then I hear. Tyrosine. Anybody know another hormone that is neither a polypeptide nor a steroid?
Melatonin.
You just released it. Huh?
Adrenaline.
Yeah, adrenaline, epinephrine. Okay? Was there an effect from it? Anybody feel the effect? Pretty quickly? So what's it behave like? Behaves like a polypeptide, fast onset, and hopefully now things have calmed down and it's okay. Fast onset, short acting. Behaves like a polypeptide, but it's not. What's it made from? Huh? Polypeptide made from tyrosine. The two neithers, epinephrine and thyroid hormone, are both made from the same precursor, tyrosine, an amino acid. Okay, but I digress. So, when we're looking at things, if we want to alter the behavior of a cell, we need to alter total activity of specific proteins. Total activity is specific activity times total amount. So hydrophilic signals, fast onset, short acting, they're the cattle prods. They are aiming at largely changing specific activity of existing protein molecules. The steroid hormones and other lipophilic signals, they're primarily looking to alter gene expression and thereby change the total amount of the total number of protein molecules in the cell. Okay, let's take our break. When we come back, we'll do the specifics. We've set the stage now. I'll talk quickly.